Hello everyone, my name is Jacob Schreiber and I'm a postdoc at the University of Washington in the Department of Genome Science. In the past I've presented my open source contributions to SciPy, but today I'm going to be talking about my research, which involves characterizing human epigenomics through imputation. So let's delve into some background. Perhaps the most important open research area in biology right now is trying to understand the basis for the diversity of human cell types and tissues. There are a lot of them. People are very familiar with a lot of the common tissues like lung, heart, skin. And while these tissues are very important and are studied themselves, they are also made up of mixtures of cell types that are distinct from each other. And so this results in a massive number of cell types and tissues with slightly different functions that are important. But differentiated tissues aren't the whole picture. There are also undifferentiated and partially differentiated stem cells that are the basis of all of our differentiated tissues, and all of those are important to study as well. Further, we've been mostly looking at adult versions of these cells when fetal versions of these, the fetal counterparts of these cells are distinct and important to understand as well. So throughout this talk, I'm going to be using the term biosample to refer to all cell types, tissues, primary cells, genetic modifications, treatments, etc. Basically, just for simplicity, I'm going to use this term biosample. So the question remains, what gives rise to what gives rise to all of these different biosamples, given that every cell in the human body has the same sequence? Well, to try to answer this, researchers have they've developed and they've performed these assays that try to measure biochemical activity along the genome. These include, say, RNA-seq that will measure transcription and gene expression, DNA synth-seq that will measure accessibility, how available is a region of the genome to, say, binding of proteins or molecules, and chromatin immunoprecipitation followed by sequencing, or ChIP-seq, that will measure the chemical modifications to the histone scaffold that DNA is wrapped around, or the binding of proteins to the genome itself. So, altogether, I'm going to refer to these phenomena, accessibility, transcription, histone modification, and protein binding, as epigenomics, even though there's a little bit of controversy over what precisely that term means. I'm just going to use the term epigenomics. So the output of one of these assays is a signal track that spans the entire genome. Here's an example of 3 signal tracks for the histone modification H3K4ME3 in 3 different human biosamples. You have IMR90, HepG2, and aorta. You'll notice on the right-hand side that there are peaks. These peaks indicate an enrichment of a certain type of activity. Here, this refers to an enrichment of this specific histone modification at this position in the genome. But the fact that there's a peak in each cell type indicates that this activity is similar from one cell type to the next. However, there are some peaks in the middle that indicate that there is more biosample-specific activity. And this biosample-specific activity is really important because understanding where there's differences in biochemistry along the genome can help us understand the molecular basis for things like the presence of cell types or, when things go wrong, disease. So these are the really important regions. Given how important these assays are, large consortia were formed that had several goals, one of which was to try to perform as many of these assays as possible. An early consortium was the Roadmap Epigenomics Mapping Consortium, and they produced a compendium in 2015 that had over 1,000 experiments in it. Here we have a dense slice from the compendium that they released, and it spanned 127 human biosamples, and they used around 24 different assays. The investigators on this project knew that they wouldn't be able to perform all of the assays. They wouldn't be able to perform all of the experiments in this grid, so they set out in a very targeted fashion. Specifically, they chose 5 assays beforehand that they knew would give very complementary sources of information, and they performed them comprehensively in all 127 biosamples. Then they had a few biosamples that were easier to grow in the lab, and so many experiments were performed in them. The result is that you have this kind of L-shaped structure where you have some biosamples that are thoroughly assayed, and you have many assays that are comprehensively performed across all the human biosamples. So the question then arises, have we characterized human epigenomics yet? Well, the answer is obviously no, because this matrix isn't filled in. There are around 3,000 potential experiments that could be performed, and they performed only around 1,000 of them. However, these 1,000 measurements are somewhat difficult for people to make sense of. If you wanted to load up maybe 5, 10, 20 tracks and look at them at the same time, you might be able to make sense of what's going on. But trying to load up 1,000 is super difficult. And if you're trying to build a computational method that used 1,000 measurements for every base pair in the genome, it would take a lot of computational power to have it scale genome-wide. So we have kind of two opposing goals. The first goal is, can we fill in this matrix? Can we just impute all of the experiments that haven't been performed? The second is, can we compress the available measurements into a low-dimensional latent representation that's easier for us to manage? If we can get these 1,000 measurements down to, say, 100, that would be an order of magnitude faster being able to work with. So we chose a method that relied on deep tensor factorization, but before we get to what that is, I'm going to introduce it by explaining first what matrix factorization is. And it seems like it's a requirement when talking about matrix factorization to talk about it in the context of the Netflix challenge. So the Netflix challenge was posed at a high level as this: can we take all of the ratings of shows that people have watched and use that to predict how they would like shows that they haven't yet watched? This was in the context of trying to recommend shows that they anticipate that viewers would like that they haven't yet watched. Well, matrix factorization is an approach that tries to learn profiles or low-dimensional representations for each viewer and for each show separately. By factorizing out these representations along the two axes of the matrix, they're able to ultimately make predictions for every element in it. So the idea here is that you want to use all of the data for each viewer in order to learn a representation of that viewer, and all of the data for each show in order to learn a representation for that show. What you get is a profile where each dimension corresponds to some quality. For example, one of the dimensions in the show might be, is this show thought-provoking? The corresponding dimension in the viewer representation might be, does this viewer like thinking? If both of those things are true, it's likely to contribute to a higher predicted rating for that show from that viewer. In reality, it's unlikely that the concepts line up that cleanly with human interpretable phenomena, but it's a good thing to keep in mind when thinking about this. So now that you have all these profiles, what do you do with them? Well, because you have separate representations for each show and for each viewer, you can use a function in order to combine them in order to make predictions. This function is typically just a simple dot product, where you take the vector representing each show and the vector representing each viewer, just take the dot product between them, and that's your resulting vector, your resulting prediction. So, how does this work? Well, you might take a viewer representation, such as me, my representation, and you might take a show representation, such as that of Gossip Girl, and you would take those, you'd feed them into the dot product, and you would make a prediction. Hopefully this model would predict that I would give that show a high rating, because as many of you know, I love Gossip Girl, and I've accordingly upvoted it on Netflix. But it's not particularly challenging for the model to to make predictions for shows that you've already watched. Let's say that you wanted to make a prediction that doesn't yet exist in this matrix. For example, you would substitute in the factors for my advisor, Bill Noble, and because Bill is an old man, he would give Gossip Girl a poor rating because he doesn't like happiness or fun. So then you can take all pairs of show representations and viewer representation and end up with a fully complete matrix with predicted ratings for every show-viewer combination. We then extend this to deep tensor factorization in a model called Avocado. It's a tensor factorization model because instead of having two dimensions like the matrix factorization, it has three dimensions: the assay axis, the biosample axis, and the genomic position axis, because you're modeling every position along the human genome at 25 base pair resolution. So you end up with these three sets of factors. Then it's a deep method because instead of using a dot product in order to combine these representations, you use a neural network. This has the convenient property that, in addition to being more accurate because it's a deep model, of course, it allows you to set different representation sizes for these three axes. And so since the genome position axis is significantly larger than the other two, we can get large representations for assays and biosamples while keeping the genomic position axis more compact. Then the profiles, the latent representations, and the weights of the neural network can be trained together using standard gradient descent at the task of predicting values within the tensor that we have. So I said that there were two problems that we were trying to address. The first was, can we impute everything, which is going to be the primary focus of this talk. And the second is, can we compress this information into an informative representation? And I thought I'd just include one slide on that because it's really interesting. And each one of these axes has resulted in a different line of future work. The first thing we did is we took known promoter regions and known enhancer regions from the genome, and we extracted their representations and projected them down using UMAP. What we found is that there seemed to be a pretty clear cluster of enhancer regions and a pretty clear cluster of promoter regions, which is useful because the two serve different biological roles. However, instead of being very distinct clusters that are far apart, there seems to be a continuum between the two. And this is consistent with modern biology, which knows that it's more complicated than simply calling something an enhancer or a promoter. Some promoters serve as enhancers for other genes. Some enhancers serve as alternate start sites for genes. So it really is more of a continuum between the two, and the fact that our model is able to pick this up is very encouraging. Then we looked at the assay representations, and there aren't really enough assays to see a lot of structure, but what we do see is that the assays that measure transcription-associated phenomena are together in one part of the plot. We have the marks that are associated with activating marks, associated with active gene expression kind of in the middle, and we have the two repressive marks at the bottom. So a little bit of evidence showing that we are learning important phenomena there. Then the biosample embeddings on the right-hand side here are very interesting. What we see is that there seems to be a pretty distinct clustering of cell type based on their anatomy type. We have blood cells in blue on the left-hand side. We have brain cells in the bottom right. We have digestive cells close to the brain cells. It's pretty interesting. And unfortunately, I don't have enough time to go deep into this. But each one of these has, when we've delved deeper, there's a really interesting story behind each one of them. So let's get back into the imputations. We started off by just visually inspecting how well it seemed like our model was doing. So here's a track of the histomodification H3K4ME3, which is what I was showing you before. Avocado is not the first imputation method. The first one for this large amount of data was Chrome-Impute, which was proposed by Jason Ernst and Manolis Kellis at MIT. And so you can see that that method does pretty well here. It seems like there are peaks in most of the right spots. Then there was Predicted, which is another method that was produced by my advisor in the past. And you can see that it seems like it does significantly better according to the mean squared error in the legend now. And then finally, there's Avocado, which is our proposed method. You might say that these look pretty similar to each other, and I would agree with you. They are generally similar to each other. But the thing that's most interesting is to look at the peak that Chrome-Impute is predicting on the very left-hand side and on the very right-hand side. What's happening here is that these aren't just random areas that Chrome-Impute is getting wrong. These are areas that are peaks in other cell types that aren't the one that we're currently making a prediction in. So basically, Chrome-Impute, and to an extent Predicted, are just getting confused about which cell types this peak belongs in. In contrast, Avocado is doing a much better job of identifying the true signal in this cell type. So looking at this, I was happy to, just hit submit on the paper and, be about my way. But my advisor thought that maybe we should go ahead and try to more comprehensively quantify the performance of these methods. So what we did is we used 6 different performance measures: the global mean squared error across all of the tracks, the mean squared error in the top 1% of observed positions, the top 1% of imputed positions, and then the mean squared error in the promoter regions, gene bodies, and enhancer regions to make sure that the signal is good in biologically relevant regions as well. We evaluated these using a 5-fold cross-validation, where we divided entire experiments into folds. And so what we did is we trained on 4 folds of the experiment and then tried to predict the held-out 5th fold. This is the more realistic evaluation setting because you're generally going to try to predict an entire experiment at a time. So we notice that Avocado does the best on 5 of these 6 metrics. The one that it doesn't do the best on is the mean squared error according to the top 1% of observed positions. So I don't have enough time to go fully into the story here, but essentially what's happening is that mean squared error in the top 1% of observed positions corresponds roughly to a notion of recall, which is that when a peak does exist, how good are you at predicting it? Whereas mean squared error 1 imp corresponds to precision, which is when you predict a peak, how good is it? How likely is it to be true? And what's happening is similar to what we saw before, which is that impute, which is that a primary source of error for these methods isn't just that they predict peaks at random locations that have no biological support. It's that at positions that are peaks in some cell types but not others, they can have difficulty distinguishing which cell types exhibit peaks and which ones don't. Impute has this behavior where it's more likely to overpredict peaks of these regions. It's more likely to overpredict the number of cell types that exhibit peaks, whereas Avocado doesn't have this problem. And so what the end result is that when Avocado predicts a peak, it's more likely to be real. It may end up missing some of these peaks, but when it predicts a peak, it's more likely to be real than Impute. But when Impute predicts a peak, you aren't sure whether or not it really belongs in this cell type or not. So then we wanted to extend this past the Roadmap Compendium to the much larger ENCODE Compendium. And this is because the Roadmap Compendium was incredibly useful, but it was missing some important phenomena. Specifically, the data set that we looked at didn't have measurements of transcription or protein binding in it, which are incredibly important. So we extended this to a data set we extracted called ENCODE 2018 Core. And instead of having 24 assays, it had 84 assays. And instead of covering 127 biosamples, it covered 400 biosamples. This was a total of 4,000 experiments, in contrast to the 1,000 experiments that are being included before. And so what we ended up doing is applying the same Avocado model as before, but we couldn't scale up the other methods to a data set of this size. So what we had to compare against was this baseline that we call the average activity baseline. This baseline is basically saying that at each position that we just, as a straw man, predict what the average of the training signal for that position is. So this has the convenient property that any improvements over the average activity baseline correspond to predicting biosample-specific signal. Because by definition, the average activity is as the average. It's going to be the same in every cell type in the test set. And so the reasoning behind using this baseline is that areas that always exhibit activity, like genes that are always expressed, are likely to be expressed in some test set. You shouldn't really get points for your model predicting that a gene that's always active is going to be active in some held out set. Likewise, if you have a region that never exhibits activity, you shouldn't really get points for predicting that that region doesn't exhibit activity in some new cell type either. So you can see the experimental signal here in blue, the Avocado imputations in green, and the average activity baseline in orange. I've highlighted a few regions where Avocado is doing significantly better than the average activity, but again, visually, the results are pretty compelling. Remember that this is a single model making all of these predictions. This isn't one model per type of activity. This is a single model that's able to make these exon-specific predictions for total RNA-seq, the broad histone peaks for H3K27 acetylation, and the very sharp punctate peaks for CTCF. One model is able to make all of these different predictions. So we then, again, comprehensively evaluated the performance using the same 6 evaluation metrics as before. What we saw is that Avocado improved significantly over the average activity baseline. With the mean squared error, it's able to reduce the error by over 20%, which is a pretty massive decrease in error given how good you saw just visually the imputations for the average activity were. So we then wanted to independently validate the quality of these imputations. What we did is we entered them into a challenge that had recently finished, the ENCODE transcription factor binding challenge. So in this challenge, you were trying to predict transcription factor binding in a genome-wide manner. And what we saw is that Avocado did significantly better than all of the teams in all but 2 of the tracks. In one of those 2 tracks, it did poorly. It was ranked number 3 out of 5. So it didn't do totally awful, but with one of the tracks, it seemed to not do great. Overall, there was incredible performance though. Like if you look at REST, the leading competitor had an average precision of 0.319, and we got that up to 0.477. So there are some caveats in this. For example, Avocado had access to thousands of tracks of epigenomic data that these competitors didn't. However, at the same time, Avocado didn't have access to genome sequence at all, whereas all of these models were based on trying to predict from genome sequence. So Avocado couldn't see things like binding motifs or anything like that, which is super important for trying to predict protein binding. So ultimately, though, if you are just trying to find the best predictions for an assay that hasn't yet been performed, you're going to want to use the track that has the best performance regardless of what the inputs of that were. And it seems pretty clear that Avocado is doing incredibly well at making these predictions, both in the context of just a simple 5-fold cross-validation and in the context of this challenge where people are trying to build state-of-the-art models. So I want to introduce real briefly some work that we just recently submitted, and this is on trying to make zero-shot imputations of transcription and protein binding in others across species. The idea here is basically that human epigenomics has been very well characterized at this point. Thousands and thousands of experiments have been performed, but the epigenomics of other species has not been characterized nearly as well. The question is, can we leverage the large amount of human data in order to make better imputations in other species? And so basically what we do is that we take the compendium for the other species, in this case mouse, and we basically just map the human data, all the human data, onto that species and expand the tensor out this way. By doing so, not only do we increase the amount of available training data, but we're able to get training data for assays that aren't seen in the mouse data set alone. And so by doing this, we're able to train assay factors and ultimately make predictions for assays in mice, even if that assay has never been performed in mouse before. We validated this by holding out some assays entirely from our model and seeing how well we could predict them. And just visually looking at these four, the results seem very compelling. That you have signal from like TAL1 and VAD21 and JUND that are very punctate, and it's getting activity there that's very specific to each one of these. But for CHD1, you have much broader activity. And again, this is a model that now hasn't seen any examples of these in mice, and it's performing incredibly well in making predictions in mouse. So, it shouldn't come as any surprise to anybody that at this point, the ENCODE project now has more imputed data than experimental data. We've been able to fill out a large chunk of the missing human data. We haven't yet uploaded our imputations for mice, but that's going to be coming soon. But you can see that already there are almost 37,000 genome-wide imputed tracks, whereas there are only 16,000 experimental data sets on this in this data set. And that's for us, they've been collecting data for several years now. So if you want the imputations, you can of course download them from the ENCODE project site. I would encourage you to read the several publications that we have on this now in order to get a better sense for all the details that I wasn't able to present in this presentation. But you can also, if you want to use it, download check out the code from GitHub. It's all there and it's all open source, and I provided some tutorials and documentation on how to use it. So to demonstrate the API real quickly, it basically follows that of scikit-learn. Avocado, in this case, is a predictor, and you can specify the various sizes of the neural network and the tensor factors. Then you load up the data. The data, in this case, is just going to be a dictionary, where the keys are the biosample assay pairs, and the values are the tracks, an array that spans the entire genome. And so by populating this dictionary basically represents the tensor data, and then you hit just .fit, and you pass in the parameters that you want to train on. It's very similar to that of Keras. And if you want to make a prediction, you just pass in the cell type in the assay you'd like to make a prediction for. So it's pretty easy to fit your own model. It's easy to download models that we already have pre-trained and make new predictions. We have a command line interface that lets you just automatically download the pre-trained model and make predictions, any prediction that you'd like. You can also add your own biosamples and assays to a pre-trained model. That we have code here that lets you do that pretty easily, so you don't have to retrain the entire model from scratch. And we show that even by just using a CPU, you can fit a new biosample and then make pretty good predictions, cell type specific predictions for that by leveraging the pre-trained model. So, as a summary, Avocado is a deep tensor factorization model that can model thousands of epigenomic experiments at the same time. In our initial work, we modeled 1,000 experiments. In some follow-up work, we modeled 4,000, and now we're modeling almost 9,000 experiments at the same time using the same model, and it scales pretty well, and it's able to be trained on a single GPU. These imputations are state-of-the-art. The learned representations that come out they encode complex biology, and they've been incredibly useful in trying to make sense of this massive amount of data that we have. The strength of this approach is that it's really flexible. We can apply it to many non-traditional imputation settings. And I showed you a little bit about how we can make zero-sod imputations across species. We've been also trying to apply it in the single-cell setting. There's just a lot that you can do in this case because we're applying a very flexible factorization model. And like I just mentioned, pre-trained models can be downloaded, used with a simple Python API. You can add your own biosamples and assays to that with only a few lines of code. So I'd like to thank my collaborators. Tim and Deeply have been very helpful in performing experiments and writing the papers, and my PIs, Jeff Billman and Bill Noble, they've been incredibly valuable in guiding me in these projects and helping me figure out what I should work on. So, thank you for your attention.